Yeah, so good afternoon, everyone. Today this is the eighth session, okay, and this is the last session before your exam for this course, NPTEL course. So in this session, we'll be talking basically about uh, the content which I've gone through. So first, we'll be talking about the Saccharomyces cerevisiae, its biology, okay, which was discussed in the this week content, and then we'll be talking about stem cells and its properties and the induced pluripotent stem cells, okay, how the induced pluripotent stem cells are made, and the applications and all those part we'll be discussing. And later we'll be discuss about the neurons, okay, the biology of the neurons and how the structural features of the neurons. So that we'll be discussing, and then uh, we'll solve some questions. So depending on the time, we will see, okay, we'll discuss all this portion. So I will share slide, and if you have any doubt regarding the previous classes, any of it, like okay, from starting to end, whatever we had discussed till now in previous classes. So if you want me to discuss something again, then we can have little discussion, okay, regarding it. You can ask or any topic you want to discuss again if you have not been clear in the previous class. Okay, so is this slide visible to all of you?
Yes, sir.
Okay. So in the previous class, we wanted to, as I told that about the signaling pathway. Okay, the signaling pathway was left, and one question we had skipped, and I told in the previous class that we'll be discussing in the next class regarding the aging and the mTOR signaling pathway and other signaling. So first, we will start by this topic. We will discuss this topic, and then we'll move to the discussion about the Saccharomyces cerevisiae and all the other process. Okay, so in the aging generally, metabolic changes are the main features. Okay, in the aging process, and certain metabolic pathways are activated and certain pathways are repressed, due to which there is a progression in the aging. If certain pathways are inactivated, then it will lead to the inhibition of the aging process. So there are four main signaling pathways discussed over here. One is this insulin-like growth factor 1 signaling pathway, that is IIS signaling. Second one is mTOR signaling pathway. Third is AMPK, and fourth one is sirtuins. Okay, so this growth hormones is secreted in response to glucose by the pancreatic somatotrophic axis by the pituitary gland. Okay, so when the glucose level will be higher, that is when there will be a higher food intake. Okay, at that time glucose level will be higher, and that glucose due to that the pituitary gland will release growth hormones, and that growth hormones will further stimulate the activation of insulin growth factor 1, IGF-1. Okay, so this IGF-1 will bind to the IGF receptor present in the cell, and it will activate the PI3K and AKT pathway. So before that, I want to orange colored portion. Okay, orange colored signaling molecules are the pro-aging molecules. Okay, so the molecules which you are seeing in the orange are pro-aging molecules. That is, this called growth hormone AKP-mTOR, which promotes the aging. And the anti-aging molecules are colored uh, in green, okay, somewhat green color. So, for example, this AMPK, SIRT1, PGC1 alpha. So this molecule as well as FOXO inhibits the aging. Okay, these are the anti-aging molecules. So when glucose level will be higher, then PI3K-AKT will be activated by IGF-1 receptor, IGF-1 binding to its receptor, and it will activate the mTOR. And mTOR promotes de aging. Okay, so this suggests that when the glucose level will be higher, aging process will be higher, okay? But in the dietary restriction condition, when the glucose level will be lower, okay, in the cells, and there will be a low glucose level, at that time, AMPK and SIRT1 will be activated. So, for example, AMPK will be regulated. AMPK senses low energy state by detecting the AMP level. Okay. So, in the high glucose condition, by the glycolysis and the citric acid cycle, it goes through the mitochondrial respiratory chain. It produces ATP. Okay. So, when glucose level will be higher, then ATP will be higher. But when there will be a low glucose level, at that time, AMP levels will be higher. Okay. And these AMP levels will be sensed by this AMPK. It is a sensor for the AMP. Okay. And this AMPK activation leads to the uh, inhibition of this mTOR molecule. So AMP will be active, mTOR will be inactive. So this will lead to inhibition of the aging. Okay, as well as AMP and SIRT1 acts through each other. Okay, crosstalks with each other, activates each other. The SIRT1 also SIRT2ins, that is SIRT1 is also known as SIRT2ins. So it also senses the low energy state by detecting high NAD plus level. So in the energy when the glucose will be higher and the citric acid cycle and the mitochondrial respiratory chain will be active. At that time, NADPH level will be higher. Okay, NADPH level, not NAD level. NAD plus level will be higher when in the low energy state. Okay, this is an oxidized form. When there will be a high energy state, this NAD will be reduced form. Okay, so at the in the low glucose concentration, high NAD plus level will activate this sirtuins. Okay, this sirtuins will send this NAD and it will also crosstalk with this AMPK and SIRT1 will then activate this PGC1. Okay, so this is a PPAR gamma co-activator 1A. Okay, this SIRT1 deacetylates and activates the PPAR gamma co-activator 1A, resulting in enhanced mitochondriogenesis. So mitochondriogenesis is the process which is present in the health, which decreases the aging. Okay, so mitochondriogenesis will be higher when in the healthy aging process. Okay, as well as the antioxidant molecules will be lower, antioxidant molecules will be higher, so that the oxidation produced, okay, oxidant molecule which is produced in the aging, that will limit its effect, okay, by producing the antioxidant. And fatty acid oxidation will also be improved in these conditions. All these parameters, such as mitochondriogenesis, antioxidant defense, improved fatty acid oxidation, it favors the healthy aging process. Okay, it will not lead to premature aging. So this is by activation of this PPR gamma coactivator 1A, so all the process will be enhanced. So the dietary restriction conditions, this AMPK, SIRT1 as well as PGC1 alpha will be activated, and this will limit the aging. Okay, it will not promote premature aging. As well as the in dietary restriction conditions, this FOXO and P10 level will also be higher. So P10 is a direct inhibitor of PI3K. So if PI3K is inactive, then the mTOR will not be active. Okay, and this will also inhibit the aging. As well as FOXO level will be higher, then it will also inhibit the aging process. Okay, so these are all the molecules. For example, this IGF-1 senses the glucose. Okay, and it activates and it targets the FOXO and activates the mTOR pathway. Okay, by PI3K negative. mTOR in turn senses high amino acid concentration. So when the glucose level will be higher, then the anabolism process will be also be higher. So anabolism process means the conversion of glucose molecule into the amino acids. Okay, and participation of this glucose into the structural, making the structural part of the protein as well as the other component. That is called anabolic process. So when the concentration will be higher, it will promote the anabolic process. And when the glucose level will be lower, it will promote the catabolic process. Catabolic process means the breakdown of glucose, okay, inside the cell when the level will be lower inside the cell. So anabolism will promote the aging, while catabolism will uh, depromote the aging. Okay, so it will repress the aging process. So this IIS and mTOR will lead to the anabolic pathway, while AMP can certainly lead to the catabolic pathway, and it is an anti-aging molecule. Okay, so in summary, like this is a summary that signal nutrient, IIS and mTOR signal nutrient abundance and anabolism. It will accelerate the aging process by down regulation and favors the longevity. Okay, Yeah, down regulation favor longevity. So if you down regulate this mTOR pathway, then you can uh, you can repress the aging process. Okay, it will favor the longevity. While this AMPK and SIRT2ins, it will up regulation health promotes the healthy aging. Okay, so in this, so from this signaling pathway, you can sense that during the dietary restriction conditions, so people generally talk that fasting is better for your health. Okay, so there are concept of intermittent fasting and which will lead to the balance in the glucose level inside the body. Okay, so intermittent fasting and all works through this principle that it will promote the antioxidant molecules so that the defense system will be stronger. And other features as well. Fatty acid oxidation will be healthier in that condition. So these are the signaling pathway through which this dietary restriction pathway occurs. Okay, the intermittent fasting process we talk about, and it will also prevents the premature aging in the person. Okay, in the individual. So this is the nutrient sensing pathway which is present inside the cell, and which is regulated by glucose and the dietary restriction pathway. So is it clear to everyone? This pathway?
Yes, sir.
Yeah. So this was a question which we wanted to which we had skipped from the previous class. Okay, I told that when we discuss this process that at that time we will solve this question. Okay. So this was the question. Pick the correct statement about the mTOR nutrient sensing pathway. So mTOR nutrient sensing pathway is an anabolic pathway first thing, and it will sense the glucose. Okay, and it accelerates the aging process. So first option is it senses the low energy state by detecting high energy plus level. Is this correct? Regarding the mTOR pathway, it senses the low energy state by detecting high energy plus level. So this is not a correct option. Okay, the high energy plus level is sensed by the sirtuins. Okay, sirtuins senses high energy plus level in the anti-aging process. It senses high amino acid concentration. So yes, it senses high amino acid concentration. Okay, yes, we discussed over here that mTOR it senses the high amino acid concentration. Okay, and its downregulation extend the longevity, or its upregulation activation extend the longevity. So mTOR downregulation will extend the longevity in various organisms. Okay, so these two options are correct. Option number second and third, both these options are correct in this case. Is this clear?
Yes.
Yeah. So now we'll be talking about the Saccharomyces cerevisiae. Okay, in this class. Uh, so as I told that Saccharomyces cerevisiae biology will be discussing and why Saccharomyces cerevisiae is used as a model organism. Okay, in studying the eukaryotic system, and what are the different experimental ways we can use the Saccharomyces cerevisiae. Okay, different experiments. So Saccharomyces cerevisiae is commonly known as the Baker's yeast or brewer's yeast. Okay, and it can ferment sugars to ethanol or and carbon dioxide ability produced by the brewing industry. So in the ethanol production, industrial process, okay, in the industrial process of ethanol production, we use the Saccharomyces cerevisiae. In that, they ferment sugars to ethanol and carbon dioxide. So the general glycolysis pathway present in the humans, I mean in the other organism, sugars are uptake and it goes through glycolysis and produce pyruvate. Okay, and that pyruvate is then taken by the uh, 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 that pyruvate goes through the convert that into acetyl CoA, and acetyl CoA goes through the citric acid cycle, and that the reducing equivalents produced from there uh, gives the ATP generation through mitochondrial respiration pathway. Okay, so this is the whole diagram. Okay, I mean whole metabolic process which occurs in the humans and other eukaryotes. But in Saccharomyces cerevisiae, the sugars instead of producing goes through the glycolysis pathway only, but instead of producing the pyruvate as an end product, it produces ethanol. Okay, and ethanol and carbon dioxide are produced, and because of the production of ethanol, this this mechanism, okay, is used in the industrial process to uh, uh, produce the alcohol and other beverages, okay, in the brewing industries. And the carbon dioxide released by the yeast is used in the baking process to raise the bread dough, giving it a light texture. So in place of ethanol, carbon dioxide is also produced, and this carbon dioxide uh, is uh, is also used in the baking process to make the dough. Okay, I mean to give the dough a texture in the baking industries to ferment dough in the during its preparation. So both these characteristics, ethanol as well as carbon dioxide production, is used as an industrial process, and it is classified as fungus also or mold. Okay, this Saccharomyces cerevisiae is called fungus or mold. It is for classification. It is included in that class, and it is a single cell eukaryote. It is a eukaryotic system. Okay, it has a all the features which eukaryotes contains, such as nuclear membrane and all those things. Okay, mitochondria, chromatin, and all. Yeast are like 5 micrometer in diameter, and it derives its name from the budding yeast, characteristic feature of bud formation during the cell division. Okay, so the Saccharomyces cerevisiae is a budding yeast, and another yeast is called fission yeast. Okay, the budding yeast name derives from the bud formation during the cell division, as we discussed previously also in the mitosis class. Okay, that their division occurs through bud formation. And another Saccharomyces family includes Saccharomyces pombe. Okay, that is called fission yeast. So they will fuse. Their division occurs by the fission. Okay, by fission process, their division occurs by separating with each other. So both these families, both these yeast are different in their mechanism. So when we talk about the cell wall of these bacteria, so cell wall contains like plasma membrane. Okay, and in the plasma membrane, this is the plasma membrane, and now in the outer part, cell wall contains these features such as manoproteins. Manoproteins are that is a glycoprotein, and this glycoprotein actually contains many receptors, which helps in binding to the target. Okay, in the manoprotein there are many receptors present which binds to the target protein. Okay, and there are sugar moieties as well, glucose moieties, by like containing beta 1,3 glucan and beta 1,6 glucan, as well as chitin is also present. Okay, chitin which is present N-acetyl glucosamine. It is a biopolymer of N-acetyl glucosamine. So this beta 1,3 glucan, beta 1,6 glucan are also there, and the linkage you can see that glucan means two glucose molecules are there, and in beta 1,3 glucan, this one and the third carbon of the adjacent glucose molecule is linked by the glycosidic linkage. Okay, and in 1,6 linkage, the first carbon and the sixth carbon, this OH group containing carbon, is linked by the glycosidic linkage. So that is how the bond formation is there, and the chitin provides the stability. Okay, the structural features of the cell wall. Chitin chitin provides the structural features, and it is biopolymer of N-acetyl glucosamine. So when you talk about the cellular morphology of the Saccharomyces cerevisiae cell, then the division process in generalized view, okay, how the cells divide through budding. So this is the process which happens that you have this actin, and this is the nucleus in blue color. Okay, and there are actin proteins, which is a filament, okay, which helps in the division. This is the microfilament. Okay, and during the division process, that is in G1 phase, this actin at the apical portion of the organism, there is a establishment of the polarization. So the septin proteins will bind to this. CDC42 will first define the apical part of the bud. Okay, the CDC42 will bind to the apical portion first. Then around that CDC42, septin will bind, and then actin will bound around it. Okay, and it defines the axis through which the cell division will occur. Okay, and further will it will then this actin will promote the migration of the CDC42 and septin outwards. Okay, outward of the cell, and around that region the bud will form. Okay, small bud will form around there. So there is a bud formation and polarization of the bud. Okay, and here you can see the septine portion. Septine ring will be formed around in the G2 phase. In the G2 phase, you can see the septine ring. And this septine ring actually helps in the release of the bud from the parent cell. Okay, the release of the daughter cell from the parent cell. So the septine rings helps in that regard. The green portion you can see. So now in the initial stage, there is was CD42, but now in the G2 phase, the CDC42 is present in the overall cell axis, cell periphery in the daughter cell. Okay, initially it was present in the apical portion of the daughter cell, but now in the G2 phase, CDC42, here you can see the yellow part, okay, yellow portion. It is present in the periphery overall. So this cells will now is ready to divide. Okay, and then the nucleus will be formed in this case. Okay, the daughter cell will form the nucleus, and then the division will occur in the mito M phase. Okay, at the end of the M phase, there will be a division, and in the division process as well, there are actin, septins as well as actin protein. Okay, the septin rings is there in the green color you can see. There are actin protein. Okay, actin protein, which will help in the division of this daughter cell from the parent cell. So this is the life cycle through which the Saccharomyces cerevisiae derives. So this is a generalized view. Okay, we are not talking about each and every individual signaling molecules. Okay, how the signaling occurs, but these are the molecules which play a part in the morphology. Okay, division process, basically. So key features of the Saccharomyces cerevisiae is like haploid budding is genome is 16 chromosome. Okay, it has 16 chromosome and it contains 12,000 kilobase per genomic DNA. Okay, so 12,000 kb of genomic DNA is there, and it contains a very high density of protein coding genes compared to humans. Okay, so protein coding genes are located very close to each other. So the density of the protein coding gene is higher. Okay, so all the protein coding genes are located very close to each other. In humans, generally, there are many introns are present. Okay, many introns are present in between the protein coding genes. So the density, although the number of protein coding genes are higher in the humans compared to the Saccharomyces cerevisiae, but the protein coding genes in the Saccharomyces cerevisiae is having the high density because the number of introns is little less. Protein coding genes are higher compared to humans, so the density will be higher in that case. Okay. And it also harbors the several dubious ORFs which are unlikely to code for proteins. Okay, open reading frames. Certain open reading frames are dubious, such as false open reading frames are there. It doesn't code for any protein, so that generally constitutes the intronic region of the Saccharomyces cerevisiae. Okay. And other non-coding genes are also there, such as tRNA, rRNA, and RNA involved in the several stages of gene regulation. So this tRNA, rRNA will help in the translation process to make the protein molecule. Okay, and other RNAs are also there, which are regulatory RNAs. And the origin of replication are termed as ARS or autonomously replicating regions. Okay, which are the starting site through which the replication will occur. Okay, and it is every 20 to 40 kb interval on each chromosome. Every 20 to 40 kb, each or ARS site will be there through which the replication would start during the cell division. And the centromere is also present, and in the Saccharomyces cerevisiae, it is known as point centromere. Okay, so the centromeres are actually short, that is 125 base pair in length only, and not surrounded by heterochromatin. Okay, so in the centromeric region, if when we talk about the eukaryotes, okay, earlier eukaryotes, such as in humans, for example, the centromeric parts contains a higher amount of heterochromatin region. Okay, it does not code for any protein coding gene because that region actually does not code for any protein. Okay, the regions are very dense. Okay, the DNA region is very dense in that region. So the it is protein coding regions, protein coding genes are not present in that region. But in centromere of the Saccharomyces cerevisiae gene, the heterochromatin regions is not present around the centromere. So protein coding gene is there. So it is bit unusual and different compared to the usual eukaryotic centromeres. And the telomeres is also is composed of the specialized DNA sequence and associated proteins. Okay, telomeres is also present, And the telomeres are DNA locus that codes for the rRNA. Okay, and the silent mating cassettes are the only region that form the heterochromatin. So there are very less number of heterochromatin regions in the Saccharomyces cerevisiae. Most of the regions are protein coding genes only. But the telomeres region, the rRNA coding region, okay, that is the rDNA locus, and the silent mating cascade. The silent mating cascade is actually the feature, okay, the genes which are responsible for the mating type of the Saccharomyces cerevisiae. So we will talk further what are the mating cassettes, okay, how the cassettes function. So these are the three main three region which doesn't code for any protein, okay, and that is termed as a heterochromated region. Apart from that, most of the genes are coding for protein. So now let's discuss about the mating type. Okay. So Saccharomyces cerevisiae exists in two mating types, that is mat A and mat alpha. Okay. So the haploid Saccharomyces cerevisiae that I am talking. Haploid Saccharomyces cerevisiae. First thing Saccharomyces cerevisiae about the Saccharomyces cerevisiae is it can remain in the haploid as well as in the diploid stage. Okay. It can survive in the haploid as well as diploid stage. So human somatic cell, for example, that are diploid only. Okay, the somatic cell, but the germ cells are haploid condition. Okay, but the Saccharomyces cerevisiae can survive in the can be present in the haploid as well as in diploid in specific condition. Okay, and there are these two mating type, mat A and mat alpha, and these A and alpha are the pheromones secreted by the specific mating type of the Saccharomyces cerevisiae cells. Okay, so these A and alpha are actually the pheromones. So A will secrete, mat A will secrete the A pheromones. Pheromones are basically the molecules, okay, the molecules which will be secreted and it attracts the opposite mating type. So mate A will secrete the A pheromones and it will attract the mate alpha. Similarly, mate alpha will secrete the mate alpha pheromones. It will attract the mate A Saccharomyces cerevisiae or the individual. Okay, so this is how the mating occurs between these Saccharomyces cerevisiae organisms. And each of these pheromones acts on the opposite mating type. And how does the pheromone signaling pathway operate? Is that these pheromones actually binds to this GTP. Okay, so for example, mate A will secrete the A pheromones. Okay, and these A pheromones will bind to the receptor present on the mate alpha. Okay, so mate alpha receptor will bind to A pheromones, and it will activate the GTP. Okay, it will activate the GTP G protein and MAPK cascade. And MAPK cascade, it will and this in will turn will activate the this in turn will activate the extracellular receptor. Okay, and activation of the G alpha from the G beta subunit, beta gamma subunit. So the GTP, as you know, that seven transmembrane protein receptor. GTP is a seven transmembrane protein receptor. Okay, and beneath the GTP there are these G protein. Okay, and G protein consists of G alpha, beta, and gamma. Okay, if you have studied this in the cellular biology, previous in the signaling pathway. So these three isoforms, okay, G alpha, beta, and gamma, are linked with each other. Okay, so if you want to activate certain signaling pathway, then this G alpha should be cleaved from this beta gamma subunit. Okay, so pheromones will bind to this extracellular receptor, and it will catalyze the dissociation of the G alpha from the rest of the protein component. And this free G beta, which is freed from this alpha subunit, activates the downstream MAPK cascade pathway. Okay, and this will lead to transcription of genes followed by morphological changes such as smooth formation. Smooth formation is one of the characteristics of the mating. Okay, after the smooth formation, the mating will occur between the mate alpha and mate A organism. So smooth formation, cell division, and fusion of these two different cell type. Okay, so these changes should be there before the mating process. So the pheromones which are released activates these morphological changes by activating the MAPK cascade through G protein. Okay, by binding to the GTP G protein receptor. So this is the mechanism through which it occurs, and this is how the mating occurs. Okay, so this is A cell. These are the alpha cells. Both will secrete the pheromones, and it will activate the and it will bind to the alternate receptor, okay, present on the alternate opposite cells, and it will derive the smooth formation. So this structure you can see that this is called the smooth formation, and it will promote the mating. So mating will produce the diploid cell, okay, A and alpha cell. Both the mating type will be there, A and alpha. And further mitosis will promote the diploid organism, okay. So after the mating, if the mitosis will occur, then it will promote the diploid organism, that is A and alpha. Whereas Whereas the single cell mitosis, if the mating does not occur, then the single cell mitosis will promote the haploid cell formation. So as I told that haploid and diploid, in both conditions, Saccharomyces cerevisiae can be present. So without mating, the cell will go into mitosis, then it will promote the haploid. After mating, if the mitosis occurs, then it will form into a diploid cell. Okay. So this is regarding the mitosis, but the diploid cell now can again convert into haploid cells again. Okay. So from the diploid, again, the haploid cell can be formed through meiosis process. Okay, meiosis and sporulation process. So this is actually also a very regulatory thing that when the generally the cell will undergo mitosis only. Okay, Saccharomyces, this budding yeast will undergo the mitosis process. It will not form the meiosis. Okay, it will not go into the meiosis. But when there is a nutrient deprivation conditions, this actually we discussed in the class where we were discussing the meiosis. Okay, if you remember that we talked about the budding yeast, and we talked that meiosis specific factor is there, okay, around there. Meiosis specific factor is there. So in the nutrient deprivation condition, generally the diploid cell, okay, this Saccharomyces budding yeast will not able to survive, and it will undergo the meiosis because of the in the meiosis the crossing over occur, okay, and in crossing over there is a chance that there will be a new organism which contains a different genome compared to the parent, and it will promote the survival, okay, by the additional characteristics which happens due to crossing over. So this we discussed in the meiosis class, and because of that only, that is the reason that meiosis it promotes the meiosis and sporulation, okay, so that during the crossing over pathway, the daughter cells are produced by which has a additional characteristics compared to parent cell, and which can have better survival capacity. Okay, so in that condition, it will form the haploid spores, and after germination, these haploid spores will be converted into individual A and the alpha cells, okay, daughter cells, which will be having the crossover product compared by the A and alpha cells diploid organism, okay, and this additional survival mechanism in the nutrient deprived condition. So this is the life cycle generally of the Saccharomyces cerevisiae, okay, and we discuss about certain meiosis specific regulation. Okay, you can go through the previous class and see in the meiosis class where we discussed that factor. Okay, so this is now we'll talking about the switching of the mating type. So what decides that mate alpha and mate A? Okay, so now this mate A, for example, this is mate A. This mate A can also be converted into mate alpha. Okay, so that is called switching of the mating type. So mate A Saccharomyces cerevisiae can be also be converted into mate alpha as cerevisiae. Okay, so how that occurs? This is a mating cassette, as we discussed. Okay, in the region that there are three heterochromatin region in the Saccharomyces cerevisiae. First is telomere, RDNA locus, and the silent mating cassette. Okay, that is a heterochromatin region. So this is the silent mating cassette which is present on the chromosome 3. Okay, and they contains like various subunits. There are homothelic mating lab, HML alpha. Okay, and there are HMR alpha, mate alpha is there. Okay, recombination enhancer is there, RE sequence, and there is centromere which is present in the between. Okay, and there is an HO endonuclease cut site. So when the organism need to switch the mating type, so in this case, this is A cell. Okay, okay, this is an A cell, mate A, and in there over here you can see HMRA. Okay, and here HML alpha. At the left side there is an alpha, and in the right side there is an A. Okay, so both these sequence are present in the blue color. So blue color is A sequence. The yellow color is an alpha sequence. So HO endonuclease will be activated by this recombination enhancer when the mating switching wants to occur. Then this RE sequence will be activated. Okay, enhancer will be activated, and it will lead to the HO endonuclease activation. Okay, HO endonuclease will be present. This HO endonuclease will cut at this site. Okay, it will cut at this site in the mate alpha, as you can see over here. It will cleave this region, and there will be a circularization. Okay. The chromosome will be arranged such that, for example, if you have want to convert the mate A into the alpha, okay, so HML alpha will be come closer to the mate A region, and after HML alpha will be coming to closer to the mate A, HO endonuclease will be present, okay, and HO endonuclease will be cleaved, and will cleave at this region, and there will be a recombination occurring. Okay, so this yellow portion region, which codes for an alpha type, it will recombine with this mate alpha. Okay, this A region, this blue color region. Okay, and here also the mate alpha will get generated after the recombination process. Okay, now this mate A will be converted into mate alpha, the yellow color you can see. And now this organism is converted into mate alpha. So the pheromones will be produced from this will be an alpha pheromone. Similarly, if you have the mate alpha, okay, instead of mate A, if you have mate alpha, then now the mate alpha need to be converted into A, for example. Then this HMRA, which is present, okay, over here, that region will come close to this alpha sequence, okay. This HMRA will come close to this alpha sequence, and after the endonuclease cut, they will exchange the DNA by recombination, okay, and then this mate alpha will be converted into mate A by using this HMRA template, okay, this region. So this is how the switching of the mating type occur in the Saccharomyces cerevisiae. So once the organism is produced, it is not fixed that the mating type will be same. They can switch the mating type uh, depending on the population which is present around them. Okay. So if there are if there are mate A population are higher, then some of the organism will convert into mate alpha, so that the mating will occur and for the daughter cells can be produced. Okay. Similarly, vice versa. Uh, it is done. So is this process clear to everyone? Is it clear, or we need to discuss more in this? No. Is this process clear to everyone?
Yes, sir.
Okay. If you have any doubts, then you can ask. Okay. So what is this? Will the final exam be in the form of MCQ and MSQ? Uh, that that information actually it is in the discussion section. Okay. The course tab you can see. Most of them, I think it will be the MCQs only. Okay. Some of the question can be MSQ type also. Okay. Some of the question as we discussed, that can be MSQ type as well. So mixed form it can be, but for that actually the windows, okay, the tab, the course tab, you can see what are the information which is present, and otherwise, if you have doubt, then you can directly contact to the course coordinator through email. Okay, she can better answer this because I don't know what type of questions it can be asked. Both type can also be possible, so you have to prepare accordingly.
Sir, even the timings is also not given in the mail. The date and the center has been confirmed. So it's given 4 noon.
The date and the center is there, but timing is not there
Yes, exactly.
in the whole ticket. I mean, you got the whole ticket.
No, no, not yet.
In the whole ticket, you will get the complete information. So you have to wait. The exam is in next week, right? So you have to wait. till Wednesday. Okay, Wednesday, Thursday, you can wait. Still, if you
Sure, sure.
don't get, then you can contact through mail. At that time, you will get all the information.
Okay. Yes, sir. Thank you.
So, as surrogacy, as I told that why it is used as a model system, eukaryotic model system. These are the few points which are due to which it is generally used as a model system. Okay, so first thing is that they have simple growth requirements. Okay, it can grow into in a simple sugar molecule. Okay, by providing it in the media, they grow rapidly, 90 minutes under optimal laboratory condition. Okay, so the life cycle time span is lower. They exist in the haploid and diploid state as we discussed. Okay, they can be easily manipulated. The manipulation is higher. The manipulation is quite easy in that region. A large database is already available in this as service genome database. Okay, HGD. So because we have the whole genomic region information available, so that we can have a clear understanding of the gene which we want to study. Okay, and apart from this, the intronic region, the heterochromatin regions are also lower. So most of the regions are euchromatin, which codes for a protein. So because of that, it can also use a eukaryotic model system. Okay, because the intronic region are lesser. So the in the less DNA sequence, most of the genes are present. So that is why we also use it eukaryotic this sequence services is the model system for them. So now we will be talking about two main techniques which is used for the for the mole bio okay, for the detection and studying the various gene expression and the transcription factor okay binding. So there are this ES2 hybrid system which is generally used by taking Saccharomyces cerevisiae as an organism to study okay by experimentally transcription factor binding to the DNA region. So in the East2 hybrid system, basically what it does it is depending on the transcription of the LAGZ gene. Okay, so LAGZ gene has a transcription factor GAL4. Okay, the GAL4 is a transcription factor which binds to the transcription site. Okay, promoter site of the LAGZ gene, and it helps in transcription of the LAGZ. So normal transcription of the LAGZ gene will induce the blue colonies. Okay, no in the lack of peron generally when laxation is active. So it will form the blue colony in the medium. Okay, by promoting. So there in the medium, generally you add the X-gal. Okay, and X-gal will be converted, will be cleaved by this laxation, and it will promote the blue colonies. And when laxation will not be active, then it will promote the white colonies. Okay, in the medium. So taking this as a selectable marker, the GAL4 promoter is used. So when GAL4 will bind to its promoter region of the laxate, so GAL4 will activate the LAGZ gene expression and it will promote the blue colonies. While when GAL4 is not binding, then it will not get the blue colony. Okay, it will promote the white colony. So GAL4 contains two domains. One is a DNA binding domain, another is a transcription activation domain. Okay, so both these domains need to be bound with each other or should be present near to each other to promote the active transcription activator for the LAGZ gene activation. Okay, so if these two domains are not close enough, then it will not lead to the transcription. So taking that into consideration, we have two proteins. So if you have, for example, protein X and another protein is protein Y. Okay, so protein X we use as a bait. Protein X will be bounded to this GAL4 DNA binding domain. Okay, so protein X is bounded to this DNA binding domain. And protein Y is bounded to the transcription activation domain. Okay, so now the transcription activation domain and the DNA binding domain of GAL4 is separated and individual protein is bounded with each subunit. Okay. So when these two proteins are in close enough with each other, that is when these two proteins interact with each other, this transcription activation domain will come closer with the DNA binding domain of the GAL4. And in that case, we will get the blue colonies by transcribing the LAGZ gene. But when the protein X is not binding with the protein Y, okay, that is in the green color, the transcription activation domain will be far away with the DNA binding domain of the GAL4. Okay, and in that condition, legs region will not be active because the transcription domain will not activate the DNA binding domain. Okay, for the transcription process to occur. So the yeast-2 hybrid system basically it's used to study the interaction of the protein X with the protein Y between the two proteins. If there is a direct interaction between the two proteins, this yeast-2 hybrid system is generally used. Okay, so this is one of the technique. Another technique which can be used to study the two protein-protein interaction is the confocal through the uh, confocal microscopy. Okay, by taking it with the two different protein by the different antibodies. Okay, through immunofluorescence technique. So in the immunofluorescence, generally what we do is that you have the cells, you first fixed it, okay? You fix the cell and you add the antibody, okay? By blocking, for first you need to fix it by the paraformaldehyde. Then you have to use a Triton X. Triton X is a detergent which will permeabilize the cell. Okay, so you use a permeabilization agent. Then you need to block the cells, okay, by using skim milk or BSA, bovine serum albumin. You block the cells of different proteins. Okay, so the region which don't not correspond to protein that will be blocked, and then you add an antibody specific to that protein you want to detect. Okay, so for example, you want to detect protein X in the cell and also protein Y. You add two different antibodies for both this protein, which will be specific for that protein. Okay, and after that, by ending the primary antibody, you add a second antibody which is tagged by the fluorophore. Okay, the second antibody will be tagged by the fluorophore. Okay, and that will give you the fluorescence by 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 exciting at a specific wavelength, it will give you the fluorescence. Okay, so by that technique can also be used. And if the two proteins are closer to each other, so for example, this protein is tagged with a red color dye, okay, red antibody, which gives a red fluorescence, and this another protein is tagged by yellow dye, okay, which gives a yellow fluorescence. So when they are closer to each other, it will give you the combination of red and yellow, that is green. Okay, the green signal will be there in the when we detect it through the confocal microscopy. Okay, and if they are far apart from each other, then red and the yellow signal will be separated through each other. Okay, a different red and yellow signal will be generating. So this is the another technique through which we can detect the interaction of the two protein whether it is close by or not, okay, by the immunofluorescence technique also. So another mechanism, I mean practical way we can use the sequencing service genome is by using the one-step PCR mediated tagging. Okay. So if there is a gene, for example, which codes for an ORF, okay, open reading frame, there is, for example, one gene, and you want to attach a GFP marker over there, okay, GFP and a selectable marker on that gene, so that you use that gene. So if this gene is transcribed, then the GFP will also be transcribed, and if the GFP will be transcribed, then you will directly get the fluorescence. So the detection will be easy in that case. Okay. But in the original genome, the GFP is not there. Okay. For GFP addition, what you can do, you have a forward primer which correspond to the GFP sequence. Okay. And similarly, you have a reverse primer which corresponds to this marker sequence. The whole this whole part need to be incorporated into this ORF. So first you have a reverse and forward primer which corresponds to that gene, and the tail region which is there, that should correspond to the region which matches with this ORF and this red color region. Okay, so in the original genome, in the C terminal, there is this red color region, okay, and the ORF region. So if you want to merge this GFP after this ORF region, so in the tail portion, the ORF sequence will be there, will be present, which match with this ORF region, okay, at this sequence. And in the reverse primer tail region, the red color portion should be there, which correspond to this region of the organism, okay, of the genome. So now the gene will be transcribed, and here the transcribed product, the PCR product, you can see that at the N terminal you have the portion which corresponds to the C terminal of this ORF, and the red region corresponds to this region. Okay, then you transform it, and then it will merge this product. Okay, this blue region will merge with this ORF region, and you will have the GFP marker integrated into the ORF site. And also this marker sequence which corresponds with this red color. So by the recombination mechanism, homologous recombination mechanism, which is present in the organism only, they will merge this GFP site onto the ORF. And by the Western blot technique or by colony PCR, you can screen for this organism. Okay, so if now when the gene will be active, this ORF will be active, then it will code for a GFP, and this GFP you can be detected by fluorescence microscopy and other techniques. Okay, you know, for easy detection of that gene of interest when it is transcribing. So you can add a certain portion of the DNA and merge it into the other part by this mechanism. Okay, this is called PCR mediated tagging process. So this technique is generally used if you want to integrate a certain genome, I mean certain molecule into the open reading frame. So is this clear to everyone? Both this technique, yeast to hybrid and PCR tagging. Okay, so apart from that, there are markers. Selectable markers are also present in this SRAVC. That is the auxotrophic markers, dominant markers, selectable markers, counter-selectable markers. Okay, so auxotrophic markers generally has a genes that are necessary for the growth of the cell under nutrient-deprived condition. That is LU2, HIS3, and URA3. Okay, so these are the genes. So in auxotrophic markers, generally the strains are grown into the nutrient-deprived condition. Okay, so this gene will not be present. This nutrient will not be present in that medium. Okay, so the organism which contains this gene, that organism will only survive in this auxotrophic medium. Okay, which does not contain the nutrient. So the organism which doesn't have this gene, they will not survive in this medium. Okay, so these three are the essential nutrients for the growth of the Saccharomyces cerevisiae. And if these genes are present, then only it will survive. And actually, these three genes are used in the cloning strategy. Okay, if you want to clone, if the clone are inserted in this gene, then that will disrupt that gene. Okay, and then the cerevisiae will not grow in the nutrient deprived condition. So these are the selectable markers also can be talked about. Okay. Another is the dominant markers. That is the genes that provide resistance to the cell against drugs, with the cell is sensitive, right, such as norciothresin and hygromycin B. So these two genes actually provide resistance against the drugs. Okay, so that is a dominant gene. So if these two genes are present, then that organism can survive the effect of it provides a resistance to that drug molecule. Okay. So the third one is a selectable marker whose expression select for the positive transformation. So as I told that these genes can also be used as selectable marker. So if you disrupt that gene, then that organism will not survive. And if the gene is present, then only the organism will survive. So that is called selectable marker. Okay. And there is also a counter selectable marker. For example, the genes whose expression kill the positive transformer. Okay. So counter selection for URA3 gene, for example, involves 5-fluorouric acid. Cells that express URA3 accumulate fluorouracil in the presence of fluorouric acid, rendering the cell inviable. Okay, so if, for example, there is two strain, okay, one strain which contains a URA3, both the strain contain URA3, but in one strain there is a clone sequence which disrupts the URA3 gene. Okay, so in one strain URA3 gene is not present, and generally the clone because of the insert DNA when used in the cloning, it disrupts the URA3. Another strain because the insert is not there, there the URA3 is active. So in counter selectable marker, what we do is that if the in the active URA3 generally, it will have the it will have the 5-fluorouracil, okay, in presence of fluorouric acid. So we will have the 5-fluorouric acid in the medium, and when URA3 gene is active, it will convert this 5-fluorouric acid into 5-fluorouracil, okay, and this 5-fluorouracil is actually is a mutagen, okay, and this five because of the activation of this 5-fluorouracil, it will promote the mutation and will make the cell inviable. Okay, so URA3 is active, then the cell will not be cloned. But when the URA3 will not no is not active, at that condition, cell will grow. Okay, because it will not convert it into 5-fluorouracil molecule. So that is called counter-selectable marker. And these whole strategies are generally used in the during the cloning. Okay, during the cloning process, when when the DNA foreign DNA is to be inserted into the vector and transform into this Saccharomyces cerevisiae. So Saccharomyces cerevisiae also used as a vector. Okay, the organism for the transformation. Generally, the for the transformation, is organism is also used. Okay, during the cloning process. So these are all the selection method through which we can select the clone between the clone and the uncloned DNA molecule. Okay, so these are the because of this marker present, we can select. Okay, so now we'll talk about the phytohormones. Okay, so let's take a small break for like 4 or 5 minutes. Okay, after that we can discuss like phytohormones and the stem cells. Okay, other stem cell process. Then we'll discuss some questions. So we'll meet after 5 minutes. So let's start. So this is regarding the in the plant cells. Okay, the phytohormones which are present. So now we'll discuss about the plant cells, the different types of hormones which are present in their functions. So this is important because these hormones actually play a role in the growth of the plant. Okay, in different processes. So there are five main types of hormones: auxin, gibberellin, cytokinin, abscisic acid, and ethylene. So the role of auxin is to promote the stem elongation and inhibits the growth of lateral buds. Example is the indolacetic acid. Okay, so lateral buds which are present, it promotes the apical budding only. Okay, the auxin will promote only the apical budding and it will inhibit the lateral bud formation and it also promotes the stem elongation. Okay, example is indolacetic acid. Gibberellins promote the stem elongation and the example is gibberellic acid. Cytokinins will promote the cell division during the initial stage. Okay, it will promote the cell division. Example is zeatin. And abscisic acid promotes the seed dormancy by inhibiting the growth of the cell. So abscisic acid will, in the dormant condition, when there is a nutrient deprived condition and when there is a very low level of nutrient as well as the uh, uh, water content when it is absent. Okay, at that time abscisic acid will be promoted. Okay, will be activated and it will inhibit the cell growth. And ethylene will promote the ripening of the fruits and crops. So these are the total phytohormones which are generally present in the plant cells. And one mechanism which is important to understand is the auxin-inducible degrowth system, which is present in the plant. So this mechanism is similar to the ubiquitin proteasome mechanism which we discussed. I mean, at the functional level, both are similar. But this mechanism actually helps in degradation of the protein molecule, okay, the target protein molecule. So in the ubiquitin system, when we discuss that, well, if you want to degrade a certain protein molecules, okay, for example, cyclins. So in that protein molecule, the ubiquitin will be tagged. Okay, and after the tagging of this ubiquitin, the proteasome system will be activated. So that ubiquitin tagged molecule will go through the proteasome system, and in this proteasome system, uh, the protein will be get degraded. Okay. Similar mechanism also include in the plant. For example, in the plant, if there are some proteins which you don't need at that time point, okay, which you want to degrade, and there is an auxin inducible degrowth system. So degrowth actually is a short amino acid sequence, okay, or a structural motifs that regulate the protein degradation. So instead of in place of ubiquitin, there is a degron, which is an amino acid that binds to the protein. Okay, and when the protein which binds to the degron is subjected to the degradation pathway only. Okay, and the function of this degron can be controlled by addition of the inducing compound. So in the ubiquitin system, actually, when ubiquitin is tagged, it will go to the proteasome system, and there is no inducing compound present. So, but in this case, when the degron is attached, okay, over there, auxin is generally used, and that auxin actually mediates the degradation process. So here you can see in this example that this is the target protein, and this is degron site. Okay, so over this degron is auxin-inducible degron molecule is attached on the target protein, and this is a degradation system, okay, which contains the STIR1, SKP1, CUL1, and RBX. But these are the protein moiety, protein component, which helps in the degradation process. So AID will only bind to this degradation mechanism, uh, uh, this degradation component when auxin will be present. Okay, so auxin will actually bridge these two protein component, AID and this degradation molecule. Okay, so the role of this protein component is to add a ubiquitin onto this AID. Okay, onto the degron. The protein component which culminates this STIR1, SKIP1, CUL1, and RBX. So this will add as a ubiquitin mediated system, and it will add a ubiquitin by E2 ubiquitin ligase. Okay, so E2 by the activation of E2 ubiquitin ligase, it will add the ubiquitin onto this AID. And this complex will only form when auxin will be present. So auxin will bridge the gap between these two protein component AID and the STIR1, and then the ubiquitin will be attached to the target protein, and then it will go to the proteasome mechanism. Okay, so this is the auxin inducible degron system that is in this process generally the when auxin will present, then only the degradation will occur. When auxin is not there, then the degradation will not be occur. That is why it's called auxin inducible. Okay, so the auxin inducible Lagrange system is easy and have various advantages over the other inducible Lagrange system. Okay, advantages is like both N and C terminal tagging is there. Auxin inducible Lagrange system. So this Lagrange which is attached to the target protein at both N and C terminal at the both the tail, it can be tagged. Otherwise, biologically silent. Okay, because you need an external stimulant for degradation, you can have a better control. Okay, over the degradation system. So inside, if there is no external stimulation, then the cell will automatically regulate the degradation system, and that might be or might not be helpful to the cell in some condition. But when certain, as this is a external stimulant is there, that is auxin, we can have a better regulation stages. Okay, and no measurable physiological changes are observed in this condition. So this is regarding the auxin-inducible Lagrange system which is present. Okay, that how it ubiquitin is tagged into that. So now we will be discussing about the stem cells and the different types of stem cells first, and then the concept of induced pluripotent stem cell, okay, and how you can generate the stem cell from somatic cell. So generation of stem cell from the somatic cell is called induced pluripotent stem cell. Okay, so we'll be discussing that and the application for that we will also discuss. So in the multicellular organism generally, at the earlier stage, rapid mitotic division of the embryonic cell which later differs into specialized status of tissues and organs. Okay, so in most of the cells, generally the differentiated cells are arrested in the G0 stage. Okay, that is, in neurons are permanently in the G0. So when tissues, in the early stage, there is a rapid mitotic division of the embryonic cells. Okay, so the embryonic cells will divide it rapidly and will form tissues and organs. And in the tissues and organs, generally, uh, the division is not that fast. Okay, in these organs. Okay, and in most of the cells, the cells are arrested, so there is no mitotic division occurring in some of the cells. But when the loss of cell due to programmed cell death has to be compensated by the formation of new cells, and that to achieve this balance, most tissue cells are the cells that can divide to replace cells that have died. Okay, so in these tissues and organs, generally the cells which are present in this condition, okay, most of the cells will be non-dividing, but when there will be a programmed cell death, that is apoptosis mechanism will be activated, or when there is an injury, At that time, that cells need to be replaced by newer cells. Okay, and there are some population of the cells, stem cells, the cells which are present, which can mitotically divide to replace that damaged cell. Okay, so in the tissues and organs. So proliferation of differentiated cell occur in some cases. For example, in some differentiated cells, that is fibroblast, although arrested in G1, can re-enter the cell cycle if required when the injury occurs, that is in response to PDGF. Okay, so when certain growth factors are there, then it will promote the fibroblast activation into from the it will promote the differentiation of the fibroblast from the G1 stage to the mitosis stage. Okay, and proliferation. Also, for example, in the endothelial cell, which lines the blood vessel. Okay, so endothelial cell can also proliferate in response to vascular endothelial growth factors. So in response to growth factors only, these cells such as fibroblast, endothelial cell, epithelial cell will be activated. Okay. So in the cancer, generally there is a lack of oxygen. Okay. In the cancer cell, there is a hypoxic environment, and you need a blood supply over there. So in the hypoxic condition, when there is a high number of cells, there will be oxygen deprived condition, and in that case, VEGF will be activated. Okay, vascular endothelial growth factors. So this cell will lead to the proliferation of endothelial cells, and due to and by that means there will be a blood vessel linings. Okay, new blood vessels will be formed, which will help in the better oxygen content of the cells. Okay, the hypoxic environment will be reduced in that sense. If there is a higher blood vessels. Through blood vessels, the exchange of oxygen will be there by the from the blood to the cell. There will be an exchange of oxygen. So that is why that is how it will reduce the oxygen level. Okay, it will increase the oxygen level in the cell by forming new blood vessels. And third one is the epithelial cell that can also proliferate in some internal organs. Okay, that is the liver regeneration process. Okay, so a certain process such as liver regeneration can occur by surgical when surgical removal is there. So epithelial cell can also proliferate. And when we talk about stem cells, so many fully differentiated cells can't divide, but in this there is subpopulation of cell called stem that can proliferate replace differentiated cells. So most of the cases, for example, in the neurons, okay, they are not dividing cells. Okay, and most of the cells, such as skin cells, and also they are not differentiating cells. Okay, so in that population, there is a stem cells which has the property to differentiate again. Okay, if there is and due to that mechanism, it can replace the older cells and proliferate and replace the differentiated cells. Okay, so the stem cells are the self-renewing cells present in the adults. Okay, and this cell can proliferate and differentiate into throughout their life. Okay, stem cells does not the division of stem cell occur in such a way that one stem cell when dividing into two cells, so uh, one cell will be remaining as a stem cell only, another cell will be formed into a daughter cell. Okay, from the one stem cell will be formed. Okay, and another daughter cell will be formed. Okay, so both the daughter cell will not be from only one stem cell. So that is how the stem cell will maintain its progeny as well. Okay, the remaining one stem cell will be converted into divided into one stem cell again. So that is how it will maintain its population as well during the division. Okay. And stem cells are present within the microenvironment termed niches that provides a signal to maintain the stem cell throughout the life. So in certain environment only the stem cells will be present. In uh, the environment in which there will be an adequate amount of growth factors and other factors which are responsible for the transcription of the stem cells are they present. In that environment only the stem cell will be there. Okay. In other stages, in other part that stem cell will not be there. If there is the transcription factor and all are not present, in that condition, the stem cell will not be present. Okay. So, for example, the cells which have a short lifespan, such as blood cells, sperms, epithelial cells, or skin, as well as the digestive tract. So, these all cells has a very short lifespan. Okay, the blood cells, for example, have a very short lifespan. It can continuously regenerate it into a newer cell. So, in that condition, you need a stem cell which can replace the older blood cells. Okay, so from the stem cell, the newer blood cells will be formed, and that can again circulate into the circulatory system. In that condition, stem cell is required. And also in the replacement of the damaged tissues, such as the nervous tissues, as the nervous system or the skeletal muscle, these stem cells are required. Okay. So this is the differentiation of the stem cells in the blood. How different types of blood cells are formed. Okay. By the hematopoietic stem cell. So from the hematopoietic stem cell, various different types of blood cells are formed. These hematopoietic stem cells are present in the bone marrow. Okay. And the differentiation of the stem cell in the bone marrow give rise to two cells, initial progenitor cells such as myeloid progenitor cells and lymphoid progenitor cells. The myeloid progenitor cells again will be differentiated into megakaryocytes, which will eventually form the platelets, uh, eosinophils, basophils, erythrocytes, monocytes, and neutrophils. Okay, and these monocytes will be further differentiated into dendritic cells and macrophages, which are the immune cells. And the lymphoid progenitor cells consist of T cells, B cells, and NK cells. And from B cells, you can get the plasma cell, which can secrete the antibodies as well as other immune mediators molecule. Okay, which mediates the immune signaling and the defense mechanism, promotes the defense mechanism by these lymphoid progenitor cells. So from a single hematopoietic stem cell, all these progenitors, all these cells are produced, which helps in the blood cells are produced, which helps in the immune process as well as normal blood circulation, such as RBCs, erythrocytes, as well as platelets, which is used during the blood clotting, as well as other immune cells are formed in the single hematopoietic stem cell. So how these stem cells are used in the medical application, for example. Okay, so if we talk about one example, one general example, how this medical application is done of the stem cell. So first you administer pre-treatment to release the stem cells from the bone marrow into the bloodstream. Okay, so you have certain antibiotics which you take. It will release the stem cell from the bone marrow into the circulatory system. Then from the bloodstream you collect the stem cells and preserve it. Okay, for later use. So during the chemotherapy treatment, when the patient is undergoing the chemotherapy, okay, at that time all the cells in the body will be destroyed. Okay, the blood cells, the immune system is down regulated. The immune cells will not be all the immune cells will be killed by this chemotherapy mechanism. So at that time, to again replace the immune system, the stem cells which were collected previously from the bloodstream, okay, are replaced again after the chemotherapy. That stem cells are replaced again. And that stem cell will then will differentiate into the progenitor cells, okay, myeloid and lymphoid progenitor cells, and eventually it will form the complete immune system by generating the B cells, T cells, and macrophages and all those immune cells, okay. And additionally, you have to take a supportive medicine to further enhance the immune system. In chemotherapy, the immune system will be down, but after infusing the stem cells which were collected earlier, again the immune system will get generated, and by help of the medicine, the immune system can get enhanced, okay, which can help in the survival after the chemotherapy. So during the chemotherapy treatment, this is how the immune system is restored after chemotherapy treatment. Okay, by collecting the stem cell earlier and then treated after injecting it again after the chemotherapy molecule. Okay, so this is the application of the uh, stem cell. Now we'll be talking about reprogramming of the differentiated cell. Okay, so that we talk about the stem cells and what are the medical applications. Now there was a study. There is another thing. Okay, which we need to discuss is that the reprogramming. So diploid cell, for example, the skin cell, which is a differentiated cell, can also be again converted into a de-differentiated cell. Okay, the totipotent cell. So uh, this cell is a skin cell. It is a somatic cell. So somatic cell again can be converted into a pluripotent cell, which can directly give rise to a another organism. Okay, uh, another organism can be generated. So that is called de-differentiation. From the somatic cell, again the embryonic cells is generated. Yes, that is called de-differentiation. Differentiation means the from the embryonic cell, somatic cell is generated. That is called differentiation. De-differentiation means the inverse. Okay, from the somatic cell, the embryonic cells are generated. That is called de-differentiation. So you can also de-differentiate the somatic cell. Okay, so this is what this one was the experiment. It is Gordon's experiment they performed in the 1962. So in this experiment, what he did was there were two frogs. Okay, frog A and B. From frog A, he took the egg. Okay, somatic the germ cell he took, that is egg. From frog B, he took the somatic cell, that is skin cell. In the egg, generally, in the egg, he destroyed the nucleus by the radiation. Okay. Now this is called enucleated egg. From the skin cell, he isolated the nucleus. So this nucleus is from the this somatic cell nucleus is a differentiated nucleus. Okay, and this nucleus will only code for a gene for the skin cell only. Okay, if you divide this cell, then it will not form other organs. Okay, it will only form the skin cell uh, in this nucleus. But when this nucleus is transplanted into the enucleated egg, this enucleated egg, instead of forming only the skin cell, it gives rise to a complete organism. Okay, so there was a reprogramming of this nucleus such that now this nucleus will code for all the factors which can give rise to the complete organism instead of only skin cell. Okay, so epigenetic regulations, that after the formation of certain organs as well as certain tissues and organs, there is an epigenetic changes in the DNA. Overall, the DNA in all the cells are same. Okay, that that overall the DNA content, the DNA base pairs in all the cells are same, but due to epigenetic changes, in some tissues, some of the genes will be inactivated by methylation and other pathways, whereas in some tissues that gene will be activated, and that will derive its function. For example, in skin cell, gene A will be active. In the neurons, gene A will be inactive. In that case, gene B will be active, and gene A will be inactive by methylation pathway and all the other things. But when nucleus is translocated into this enucleated egg, then these epigenetic changes are altered. Okay, and now the epigenetic changes will be such that it will derive the whole organism. Okay, instead of only providing a single cell. That was one of the experiment, and this is actually a Nobel Prize winning experiment. For that, he got the Nobel Prize for that. And by using the same mechanism, we can from this discovery actually the field of this transplantation, okay, and of this field was emerged. Okay, induced pluripotent stem cells and all those fields was derived by taking help from this experiment only. And there are many medical applications. Okay, I mean for this discovery. Although it is illegal for in humans. Okay, for example, now currently we have the CRISPR-Cas9 system. So uh, 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 in that case, if you want to mutate certain gene, you can mutate and have a altogether different function. So in the smaller organism for research purpose you can do. Okay, but it is illegal for it is unethical basically for higher organisms. Okay, for humans in all those cases. From this study, this was a preliminary study which was done. Okay, so this enucleated generally, this de-differentiation occurs by reprogramming, which involves the epigenetic changes. Okay, there is an alteration in the DNA methylation pattern. DNA methylation will be altered. Histone H1 will be replaced by oocyte-specific histone H1 variant. Okay, and histone H3 will also be replaced by another histone H3 isoform, which is used at the developmental stage. So this was the Okay, and it actually the embryonic stem cells. The embryonic stem cell were first cultured from the mouse embryos. Okay, so from the embryonic stem cell are totally pluripotent stem cell. So from the embryonic stem cells, many other different cells can be produced. Okay, different tissue types can be produced. So embryonic stem cells are cells from the inner cell mass of the blastocyst. So blastocyst is the initial stage of cell division when there is a number of cells are when there is initial stage of division. The blastocyst division occur. Okay, in the inner cell mass. And These cells are generally pluripotent cells. Okay, it is a pluripotent cells. It is not a totipotent. There are two terminologies: totipotent and pluripotent. Okay, totipotent means a single cell can be derived, can form into a all the cell types in the organism. Pluripotent means all the cell types will be formed instead of, but some of them will not be formed. So embryonic stem cell will not form the extra embryonic tissue such as placenta. Okay, so placenta will not form by this embryonic stem cell. Apart from that, other organs and tissues can be formed. So that is why it is called pluripotent stem cell. And these cells, embryonic stem cell, when given with a certain stimulation stimulant, okay, certain factors when it is provided in the medium, it can form different types of cells such as endodermal, cartilage, as well as neuron-like cells. Okay, and these are by different signaling pathways and growth factors. You can control the growth of the endothelial embryonic stem cells, and you can convert it into various other cell types as you wish. Okay, so here is an example that blastocyst. There is a blastocyst that is an inner cell mass. Okay, during the early stage of cell division, you take out this blastocyst, and this blastocyst actually will encode for various different cells such as ectoderm, mesoderm, endoderm. Okay, as well as germ cells will be formed from this blastocyst only. So you multiply the blastocyst first so that you will have a multiple cell number for this blastocyst. And as this is a pluripotent cell, it will form the ectoderm, mesoderm, endoderm, as well as germ cell by giving a specific stimulation. Okay. So for ectoderm, there will be a different stimulant which is used. Mesoderm, there will be a different stimulant which is used. Based on the requirement, for example, if you want to have muscle cells in this blastocyst, you add a stimulant which will promote the growth of the muscle cell. Okay, only. And for example, in neuronal cells, you can have a that stimulant. So from ectoderm, you can get neuronal cell as well as pigmented cells, okay, such as melanin and all those cells which are present in the pigmentation. From mesoderm, you can get muscle cells, blood cells, as well as kidney cells. From endoderm, you can get beta cells, lung cells, pancreatic cells, and from germ cells, you can get egg and sperms. Okay, so these are the different types of cells you can get from a single blastocyst depending on the stimulation which is present. And you can also culture these blastocysts, okay, and promote the growth of these cells. And you can have the possibility of clinical transplantation, okay, by using these cells. For example, in certain conditions, certain humans, for example, muscle cell is not there, then you can grow this endothelial, stimulate this endothelial cell, and promote the muscle cell growth in that organism, in that humans, okay, population. So you can use that. Now, so that was regarding the blastocyst. Okay, when you take the blastocyst, okay, that is a pluripotent cell. You can extract that blastocyst cell, embryonic stem cell, and you can convert into different organs. But there is also another concept that is an induced pluripotent stem cell. So the already differentiated cells, such as fibroblasts, that can also be converted into a stem cell, pluripotent stem cells, which has the same characteristics as that of embryonic cells. So this is the de-differentiation pathway which we are talking. Okay. The differentiation cells, that is a somatic cell, can also be converted into a stem cell by using a different sets of transcription regulators such as OC4, SOX2, KLF, and CMYC. These are the four transcription regulators which, when expressed, converts the somatic cell, that is fibroblast, into a induced pluripotent stem cell. And these cells actually can differentiate into various types of cells such as this chondroblast, osteoblast, as well as neuroblast. Okay, the bone cells, the neuron cells, as well as the cartilage. Okay, so you can also redifferentiate or reprogram the somatic cell into a stem cells by using these four transcription regulators. And this induced pluripotent stem cell formed can divide continuously, okay, indefinitely into the culture. Okay, by using a different growth factors and signaling protein, you can induce into a different cell types depending on your interest. So this was also a Nobel Prize winning experiment, okay, by the Yamanaka in 2006. They did this experiment, Takahashi and Yamanaka. So from the induced pluripotent stem cells, for example, you can generate the whole organs also. Okay. For example, cardiomyocytes, whole cells and tissues can also be generated from this induced pluripotent stem cells. For example, here you can see that there is an induced pluripotent stem cells, and you have the signaling pathway on, that is WNT signaling, that will convert into a cardiac mesoderm. Okay. Then this cardiac mesoderm will be converted into cardiac progenitor cells and cardiac myocytes. So cardiomyocytes is a present in the heart tissues. Okay, heart tissue generally present as a cardiomyocytes, which helps in the beating process. Okay, the rhythmic contraction and relaxation happens due to cardiomyocytes, and this is how it can be regenerated. So during the transplantation and regeneration process, this mechanism can be used to regenerate the mature cardiomyocytes. Okay, by using the induced pluripotent stem cell mechanism. So currently, because this is unethical stage. Okay, this is not a in this research. I mean, there is also a disadvantage associated with this induced pluripotent stem cells as well. Okay, so as a result, it is not an ethical way to generate the organs, but for research purpose, we can use this model. Okay, there is several disadvantages also associated with this mechanism. Okay, so due to that, there is unethical. For example, in CRISPR-Cas9 also, you have a beneficial function, so for disease curing function, but you can also have you can also have a downside as well. Okay, it can also be used unethically to produce a altogether a different kind of organism. So that genetic manipulation is generally unethical, so, as is this case with this system as well. So there are various consequences also, but in some case it can be used as a therapeutic strategy. Okay. So the induced pluripotent stem cells can also be used in the detection of the corrected neurons as well as mutagenic neurons. Okay, and for example, if a patient is having mutation, then you can correct it by CRISPR-Cas9, and you have a correct neuron, and you can detect by comparing with the patient neuron and the corrected neuron. You can compare what are the genes level which have changed. Okay, the mutation, and you can rescue the disease by knowing at what site and what function generally this mutation carries, and you can alter that function by the mutagenesis by the drug screening process in drug discovery. So this is a in therapeutic strategy also these pluripotent stem cells are generally used. So another concept is regarding the neurons. Okay, nervous system. So nervous system actually has a two main thing. This was all regarding the stem cells. Okay, induced pluripotent stem cells and embryonic stem cell we discussed in a certain part. The next part is the nervous system and the neurons, which we want to discuss. Okay. So there are the central nervous system and peripheral nervous system, two types of nervous system which are present. In central nervous system, generally the brain and a spinal cord is there. In peripheral nervous system, it's a sensory nervous system, okay, which carries information to the brain and the spinal cord and the motors, okay. It carries information from the spinal cord to the other parts of the body. And neurons are generally the main cells which are present in the nervous system, okay, when it conducts the or it maintains the communication through generation of action potential. Okay, changing action potential is implied by changing in charge from the positive to negative direction. Okay, in the polarized way, you conduct the transmission signals. There are three main categories of neurons: sensory neurons, motor neurons, and interneurons. The sensory neurons are response to the stimulant that transport to the central nervous system. Okay, sensory neurons transport to the central nervous system. Motor neurons collects the information from the central nervous system and it transmits the information to the other organs. Okay, and there are also interneurons which are receives information from the sensory neurons and integrates into the other neurons. So interneurons will collect the signals from the central nervous system and it will pass on this message to the other neurons, adjacent neurons. Okay, that is called interneuron pathway. And neuron anatomy, so this is a single neuron, and here you can see there are dendrites present. Okay, dendrites are the extension of the cell body, which are linked with the other dendrites onto the neurons. And soma is there, which is a cell body, with the nucleus is also present, and there is an axonic region. Okay, so these axonic regions contains the different type of cells. Okay, such as Schwann cells. Schwann cells are there, which helps in the conduction, okay, as well as myelin sheet is there. So this myelin sheet as well as Schwann cells helps in the faster transmission of neuronic impulses from the dendrites to the axon. Okay, so at the front part, the dendrites are present. At the end, there is an axon terminal. So this axon terminal will bind to another dendrites. Okay, and that is how it will pass the information from one neurons to another. The dendrites generally will take up the signals from these axon terminals and pass on to the information to the neurons through this axonic region. Okay, and there are also certain microtubules which are present, such as this tau proteins. Okay, axons have tau proteins, whereas dendrites will have a map2 protein. So these are the microtubules which are present inside the cytoskeleton in the neurons, which will help microtubules and the neurofilaments which are there in the axons and dendrites, which will help in the support system of the axons in the neuron dendrite. Here you can see the myelinated motor neurons of the peripheral nervous system. So between the two internodes, so these are the internodes. Internodes are generally protected by the Schwann cells. Okay, the Schwann cells are there and the myelin sheets are there. Okay, so internode are protected by the Schwann cells, and the node region is not protected over there. Okay, so this node region is also called the nodes of Ranvier. Okay, so between adjacent Schwann cells are uninsulated region. So this region is uninsulated, and it is called the nodes of Ranvier. Okay, and the internals are protected by this basal lamina as well as the myelin sheet, which will help in the faster transmission of the signal action potential from the cell body to the axon terminals. Okay, so myelin sheet generally help in the protection of the neurons as well as the faster transmission of the signaling of the action potential. In action potential, as you know, that it is a sudden reversal of the voltage between the cell membrane. So how the action potential will be generated? For example, in the initial stage, if the plus positive charge is higher, then the action potential will lead to the depletion of this positive charge and increase in the negative charge. Okay, so this charge reversal will be there, and this will help in the due to this reversal of the voltage, it will generate the action potential. Okay, and this is temporary. Okay, the negative charge again will be down regulated, and positive charge will increase. So this reversal of this charge will lead to the action potential generation within the neurons. And this change in the action potential will generate the electrical impulse, which will sweep to the neurons. Okay, and that will promote the signaling. For example, here you can see that from the dendrite there is a generation of action potential, and it reaches to the axon. Okay, and from the axon terminal there is a synapsis. Okay, synaptic cleft is there. So between the dendrite and the axon, there is a synaptic cleft is there, and the synaptic role of the synaptic cleft is to release certain neurochemicals which binds to the dendrites of the adjacent neurons. Okay, postsynaptic neurons. This will again lead to the signaling pathway activation and the action potential generation in the postsynaptic neurons. Okay, so the action potential which is generated it will activate this signaling molecule, and signaling molecule will burn off from this presynaptic neuron. Okay, from the presynaptic neuron, the signaling molecule will budding off. Okay, it will be released from the signaling molecule due to generation of this action potential, and it will bind to the receptor present on the postsynaptic neuron, and it will activate the signaling mechanism again, which will generate the action potential in the adjacent postsynaptic neuron. Okay, that is how the signal is relayed by these neurochemicals. So this is a detailed view of how it is done. Okay. So calcium ion is generally there. Okay. By release of this calcium ion, it will bind to this receptor. It will activate the receptor pathway. And there are various neurochemicals which are present. Okay. Acetylcholine, dopamine, serotonin, and all. So depending on their release, it will mediate the signaling pathways in the adjacent neurons, and further neurochemical transmission will occur. So these are the four types of neurotransmitters which are present generally. Okay. Acetylcholine, which is released at the neuromuscular junction between the neuron and the muscular junction. Okay, in the presynaptic cleft, in the synaptic cleft it is released, and it can be excitatory as well as inhibitory, depending on the receptor which is present. Another is serotonin, which is released in the areas of brain and spinal cord. Okay, the neurons which are present between the brain and spinal cord, over there the serotonin will be released, and it regulates the moods, sleep cycle, and the appetite. Third is the dopamine, which is released in the areas of the brain and the part of peripheral nervous system. Okay, and it plays a role in the emotions, the dopamine. Fourth one is glutamate, which it is an excitatory neurotransmitter in the brain. Okay, when the neurons are excited, it releases the glutamate, and it will activate the adjacent postsynaptic neurons in this case. So these are the four types of neurotransmitters which are present in general when we talk about the neurotransmitter. So you have any doubt till here? You have any doubt
No,
in
sir.
this? Okay. So now we'll be discussing some questions. So these are some previous year, previous year model questions which are there uh, for the regarding this content which we discussed. Okay, so you can practice questions. Some practice questions are there regarding the content we discussed today. The first question is the pick the correct statement about the stem cells. The stem cells are self-renewing cells. This is the correct. They can only proliferate in a specific age bracket. No, this is not correct. They can proliferate at any age. Okay, it is not a specific age. They give rise to two daughter cells that remain as a stem cell. No, one stem cell will divide into one daughter cells and another stem cell. Okay, from one stem cell, two daughter cell will not be formed. So these two are incorrect. So option one is the correct. That is, stem cells are self-renewing cells. The second option, second question is which of the following can be used to arrest MET alpha type of S services cells in the G1 phase state? Okay, so here you have three molecule, uh, and it is shown, it is present that it is told that from these three molecule, uh, which of the molecule will arrest the MET alpha cells in the G1 phase. So when the cells will be arrested in the G1 phase, the mating will not occur. Okay, the mating will not occur, and the division also mitosis process will also not occur. Okay, so alpha factor, alpha factor will promote the mating. Okay, it is a pheromones which will promote the mating by promoting the mitosis process. Okay, and formation of the smooth. Hydroxyurea and ocadazole. These two are also not there. Okay, because hydroxyurea and nocadazole is basically the inhibitor of the microfilaments. Okay, and these two molecule will also not inhibit the arrest the MET alpha cells in the G1 phase. Okay, so none of the option is correct. So the fourth option will be correct in this case. Okay. Third question is which of the following can correctly define the oxotrophic marker? So oxotrophic marker, as we discussed, that oxotrophic markers are such markers which are present in the cells which are grown in the nutrient deprived condition. Okay. The genes which are essential in the nutrient deprived condition, that is the genes which code for the essential nutrient. Okay, that is called oxotrophic marker. So first option is gene necessary for the cell to grow in the essential nutrient deprived condition. Yes, this is correct. Gene necessary to grow in the presence of drugs like hygromycin. No, hygromycin. This is not a correct option. Gene necessary for the cells to grow under non-essential deprived condition. No. The oxytrophic strength will grow in the essential nutrient deprived, not non-essential. So option A would be correct in this case. The non-pigmented plastids are called leucoplast, chloroplast, chromoplast, or xyloplast. Okay. So the non-pigmented plast, this is the question from the plant cell. Okay. If you have gone through the lecture, then you can answer this. We have not discussed today regarding this, but chloroplast contains a green pigment. Okay. Chromoplast and xyloplast also is a pigmented plastid. Also contains a pigment molecule. Okay. But leucoplast is not. It is a non-pigmented plastid. So leucoplast is correct. So after this class, you can go through what is leucoplast. Okay, how it functions. So you can go through them. In the budding yeast genome, which of the following have a heterochromatin-like organism? So in the budding yeast, as we talked, that there are three main heterochromatin region. First one was telomere, another one was RDNA locus, the third one was silent mating cassette. Okay, so these were the three heterochromatin region in the budding yeast. So let's see the option. First one is the RDNA locus. Yes, this is the correct. Centromere, no. Centromere is not a correct option. The centromere contains a euchromatic, it does not contain heterochromatin region. SMC gene locus, SMC gene locus and the telomere. So telomere is correct, SMC gene locus. So this option actually is also correct, okay, but I don't know why only the two option is given. SMC stands for silent mating cassette, or I don't know this SMC whether stands for the SMC protein which we discussed, okay, earlier in the mitosis process. So is that this SMC or the silent mating cassette? So if this is a silent mating cassette, then this option is correct. Otherwise, the SMC which we discussed, which helps in the chromatin and condensate, for which consider the chromatin and condensate, then this option is incorrect. Okay, so this option is little doubtful. Okay, otherwise it's RDNA locus and telomere option. Both these two options are correct in this case. In the plant system, the coaxial membrane channel that links the cytoplasm between cell to cell communication is called plasmodesmata. Okay. So in the plant cell, if you have studied, then there are two cells, and in between there is a channel through which the nutrient can pass through each other. Okay, so that that channel is called plasmodesmata. Okay, ability of the cell, which is the ability of the cell, which an ability to produce a complete organism is called. So here it is called complete organism. Okay, totipotent, pluripotent, multipotent, and unipotent. So in pluripotent, as we discussed, that it will not form complete organism except for a single cell in the embryonic stem cell, for example. It doesn't it doesn't express the placenta. Okay, so it will not form a complete organism. So pluripotent will not be there. Only in the totipotent, if the cell is totipotent, then it will form a complete organism, including the placenta. Okay, so embryonic stem cell is pluripotent cell, not totipotent. So this ability to produce a complete organism is termed as totipotency. Okay, so option A is correct in this case. So a fibroblast can be reprogrammed to cells similar to embryonic stem cells by co-expression of which are the following transcription factors. This we discussed, okay? The four types of transcription factor which was needed. So OCT4, SOX2, KLF4, and MYC. All these transcription factors are needed. Conversion of these fibroblasts into an embryonic stem cell. Okay, so all of the above will be the correct option in this case. Here we discussed that fibroblasts will be converted into induced pluripotent stem cells by these four transcription factors. So option 9, which of the following neuron transmit information between the parts of the central nervous system? Okay. So between the parts of the central nervous system, there is an interneurons. Okay. Sensory neuron generally takes the information from the in the brain. Okay. Central nervous system. Okay. In the brain. Motor neurons transmit information from the brain to the other motor proteins, other muscles and all proteins. Okay. Muscle cells, neurons. But interneurons actually communicate between the parts of the central nervous system. So this C option is correct in this case. The neurotransmitter that is released in the neuromuscular junction. This we seen. Okay, the acetylcholine. Acetylcholine is released in the neuromuscular junction, and it helps in the uh, transmission of information from the brain to the muscle cells, okay, muscle neurons. So acetylcholine is the correct option. Yes, yes. So now these are the any question? Anyone have any question?
Sir, can you please talk about multipotent?
Yeah. So this multipotent. Multipotent means a specific type of cell. Okay. So in totipotent, what we discuss in totipotent, all the cells, including the inner cell mass that is blastocyst as well as the placenta. Okay. All the cells will be encoded by this totipotent cell. In the pluripotent, placenta will not be produced. Only the blastocyst cell, and in that case, mesoderm, ectoderm, endoderm, sperm cell, the germ cells, all these cells will be produced except the placenta. That is called pluripotent. Multipotent means only a single subset of the cells will be produced. For example, in endoderm. So in endoderm there are different cell types such as blood cells, okay, and other cells which are there. Okay, in the ectoderm, for example, if we talk that there are neuronal cells, there are epithelial cells, so they are coded by the ectoderm. So multipotent means only a single subset of cell which are produced. So the ectoderm is called a multipotent cell. So ectoderm will not code for a cells which is coded by the endoderm cells. Okay, okay. So for example, in endoderm there are lung cells and all those cells. So ectoderm will not code for the lung cells and all. Okay, so that is called multipotency. Okay. Some of the subsets of cell will be coded, but not all the cells will be coded, will be expressed. And unipotent means a single subset. For example, now cell will be skin cell will be forming only skin cell, not neuron cells. So that is called unipotent. Okay.
Yes.
So is it clear?
Yes.
Yeah. So each and every part has an example. Okay. For pluripotency, you have the embryonic stem cell. For multipotent example, you have endoderms, endoderm, mesoderm. Okay. The cells coding for that. And in unipotent, you can have any cell type which have a single which code for a single tissue. So cells only. So these are the example. These are the questions for previous week assignment. You have already uploaded that assignment. Okay, applied for that assignment, and we can discuss this from previous class, previous year. I mean previous week. These were the questions. First one was rapamycin inhibits mTOR kinase. Okay, so mTOR actually stands for mammalian target of rapamycin. Okay, so these are the full form. So mTOR rapamycin can inhibit the mTOR kinase, and mTOR actually helps in the aging process as we discussed. Okay, starting with the lecture. If you have a so in this case, if you have a rapamycin, then it will limit the mTOR kinase, and you can delimit the aging process in that case. But it is not a simple way. Rapamycin has other consequences as well. Okay, mTOR instead of on in the case of aging, there are also other signaling pathways which is activated by the mTOR. Okay, which is beneficial. So you cannot directly target mTOR. Okay, so option one is correct. Cell swelling, membrane rupture, inflammation can accompany which option? Necrosis. Okay. So necrosis generally the membrane rupture was there, inflammation would be get produced and cell swelling will be there. But in apoptosis, generally in apoptosis the membrane blabbing was there and membrane was not getting ruptured. Okay, the membrane blabbing was getting occurred. Okay, and the cell was shrinking, not swelling. Okay, in the necrosis generally the cells are shrink, swell, but in apoptosis cell gets shrinked. Okay, the area get reduced, and in apoptosis the inflammation is not produced. Okay, and senescence is a growth related stage. Okay, in that case inflammation and all those things is not we don't have the inflammation and all in the senescence, so necrosis option is correct in this case. Which of the following is a pro-apoptotic gene? So there are pro-apoptotic and anti-apoptotic. Okay, so BCL2 was an anti-apoptotic gene if you remember the earlier lecture. Okay, so BCL2 was an anti-apoptotic gene which was inhibiting the apoptosis, MCL1 is also an anti-apoptotic gene. It is not a significant, it does not play a significant function, but is one of the anti-apoptotic gene. And also A1. A1 is also an anti-apoptotic gene. BAC and BAX were the pro-apoptotic gene. Okay. BAX as well as other pro-apoptotic genes are also there. NOXA, PUMA, and all those things. Okay. So those are all pro-apoptotic genes. So you need to remember what are the anti-apoptotic gene and what are pro-apoptotic gene from that table which we discussed in the previous class. Okay. Which of the following ions plays a crucial role in control of the various forms of cell death, such as necrosis, apoptosis, and autophagy? So calcium is the correct option. So calcium ions actually plays a very important role in the maintaining the cellular physiology. Okay, through calcium, many signaling pathways are activated, and necrosis, apoptosis, as well as autophagy are also regulated by calcium. Okay, and in the mitochondrial system, okay, in the mitochondrial system also, calcium is generally plays an important role. Okay, release of calcium by the mitochondria actually promotes this necrosis and apoptosis and autophagy pathway. Okay, the calcium because calcium is used as a cofactor by many of the enzymes in the cells. So because of that, when there is a high level of calcium, that enzyme will be get activated and it will lead to the formation process of the necrosis and all. So calcium is the correct option in this case. Okay. PET scan uses which of the following properties of cancer cells. So PET scan we discussed in the previous class that the PET scan generally the cancer cell consumes high level of glucose. Okay. And in the PET scan, generally the glucose levels are detected. If you have large quantities of glucose, then it can be detected by the PET scanning method. PET scanning stands for positron emission tomography. Okay, so radioactive glucose is generally used, and that radioactive glucose is uptaken by this cancer cell, and that can be detected by the PET scan method. Okay, so option one is correct. That is cancer cell input large quantities of glucose. So radioactive glucose will be uptaken, and that we can detect by brain scanning image. Okay, in the brain, we can detect the level of glucose. Apart from brain and the other organs, we can also detect radioactive emission. Okay, detecting the radioactivity. Which of the following is not a hallmark for cancer cell? Not a hallmark. Okay, so resisting cell death. This is the one of the hallmark. Enabling replicative immortality. Okay, so yeah, so cancer cell is an immortal cells. Okay, so this is also correct option. Genomic instability is there in the cancer cell. That is why it induce the cancer progression. Okay, because of genomic instability. So all these three options are correct. So none of the above will be the correct option. I mean, because all these three are the properties of the cancer cell, and you have to suggest which is not. Okay, so none of the above is the correct option. State of stable cell cycle arrest in response to the diverse stresses or intracellular damage is termed as cell death, senescence, aging, none of the above. So senescence is the correct option, okay, when there is a nutrient deprived condition or when there are various stress observed or when there is an intracellular damage, then the cell will undergo senescence, okay, the replication and the replication and the division will be inhibited in that condition. So senescence is the cell cycle arrest, okay, condition. This option pick the correct order of the steps in the performing 3C. Okay, so 3C technique. This is actually the previous to previous week question which is present over here. So 3C technique basically was to identify the two histones. Okay, which was interacting. We discussed in previous to previous class that when two histones are generally interacting. Okay, two chromatin regions are two histones, when the two histones are far apart, you want to see that whether it is interacting directly or not. So first you cross-link that histone, okay? You cross-link that two histone by each other. So and then you restriction digestion the chromatin. So chromatin region will be digested. Only the histone region will be there. And you ligate that two histone. You purify that DNA which is encoded by that histone, and then you and pair enrich. So 3C in the 3C technique when we discussed about that how the when the two genome, okay, the two DNA region when it is far apart from each other, then how it is simultaneously regulated. Okay, so this one genomic region will directly regulate or will act as an enhancer for another gene. Okay. If they are far apart from each other, then how can it be regulated? Okay. So this 3C technique is used generally because the histones, due to when because of the histones, okay, so these histones will interact with each other. Okay, it will because of the histone present in that DNA. These two histones will be come closer to each other, and that is how the two DNA region will interact with each other. Okay, so the 3C technique is generally used to identify that region. Okay, the DNA regions are far apart, and then but it is encoding with each other. So can go through the 3C technique from the previous lecture we have already discussed. So in this case, this option is correct. Okay. First, you need to cross-link the two histone. Then you need to digest chromatin. Then you have to ligate the two histone. Okay. Again, then you have to purify the DNA. Then you ligate the DNA basically, which is encoded by that histone, which is wrapped by that histone. You purify the DNA, and then you have the pair and reads. Okay. You read the pair, which is ligated. Right? What are the DNA region which correspond to that, which is encoded by that histone. So it is the 3C technique. The process of spreading of tumors from the site of origin to other sites of the body through the lymphatic and circulatory system is termed as metastasis. Okay, so metastasis is generally the cancer cell migration from the one organs to another tissue organs. Okay, so it's called metastasis. Which of the following virus can cause cancer? Papilloma virus, human HPV, EBV. Okay, and all of the above. So all these three virus. Okay, papilloma virus as well as HPV and well as EBV. All of these three viruses can cause the cancer, this cancer causing virus. So all of the above will be the correct option in this case. Okay, so this is all regarding the today's class and like for this session, for this course. Okay, this was the last lecture. And if you have any doubt, okay, from other classes, if you have any doubt, because this is the last lecture before your exams. So if you want some discussion in other topic, we can have 5 to 10 minute discussion on that. Okay, so if you don't have anything to discuss or no, is there, if you don't have any doubt, then we can end this session. Okay. Okay. Thank you everyone for joining, and all
Thank
the
you so
best for
much, sir.
your all the best for your exams.
Thank you.
Thank you, sir.
Yeah.
Thank you, sir.